Welcome to IQriously. Get comfortable. Take a slow breath. That breath you just took. The oxygen in it was made by a plant. Not a specific plant you know. Not the tree outside your window or the houseplant on the shelf. But a plant or an alga or a cyanobacterium somewhere in the world captured sunlight, split water, and released oxygen into the atmosphere as a waste product. The oxygen you are breathing right now was made by photosynthesis. Every molecule of it. Photosynthesis is the process by which plants and certain other organisms convert light energy into chemical energy. It is the foundation of almost all life on Earth. It produced the oxygen that fills our atmosphere. It captures the energy from the sun that powers almost every food chain on the planet. And it happens inside a structure so small that you could fit 1,000 of them across the width of a human hair. Tonight, we are going to understand it. Not just the equation, though we will get to that, but the actual extraordinary step-by-step process by which a leaf turns sunlight into sugar. There is nothing you need to do. Nowhere you need to be. Just listen. Let's begin with light. Sunlight is electromagnetic radiation. It travels from the sun to the earth in waves at the speed of light, covering 150 million kilometers in about 8 minutes. The energy in sunlight comes in discrete packets called photons. Different wavelengths of light carry different amounts of energy. Ultraviolet light, shorter wavelength, carries more energy than visible light. Infrared light, longer wavelength, carries less. Within the visible spectrum, violet and blue light carry more energy than red light. This matters for photosynthesis, because photosynthesis begins with the capture of photons. And not just any photons, specific wavelengths. Inside every green plant cell are small structures called chloroplasts. The chloroplast is the machine of photosynthesis. It is a remarkable little organelle enclosed in 2 membranes, filled with a fluid called the stroma, and containing within it a system of tightly folded internal membranes called thylakoids. The thylakoids are stacked in piles called grana singula granum. And embedded in those thylakoid membranes are the molecules that do the first essential job of photosynthesis, absorbing light. The most important light-absorbing molecule in plants is chlorophyll. There are 2 main types, chlorophyll A and chlorophyll B. Both are large, complex organic molecules with a flat ring structure at their center that contains a magnesium atom. That ring structure is what makes chlorophyll a light absorber. It can capture photons of specific wavelengths and use their energy to excite electrons, to push electrons to higher energy levels. Chlorophyll A absorbs light most strongly in 2 wavelength ranges: blue-violet light at around 430 nanometers and red light at around 680 nanometers. Chlorophyll B absorbs different wavelengths, blue light at around 480 nanometers and red light at around 640 nanometers. What chlorophyll does not absorb is green light. Green light is reflected. This is why leaves are green. Not because chlorophyll is green exactly, but because green is the part of the spectrum it does not absorb and therefore bounces back into your eyes. The chlorophyll is absorbing the blue and the red, throwing the green away. Leaves are the color of light they cannot use. That is a strangely poetic fact. Alongside chlorophyll, plants contain accessory pigments, carotenoids, and xanthophylls. These absorb different wavelengths of light, particularly the yellow and orange range that chlorophyll misses. They also serve a protective function, helping to dissipate excess light energy that might otherwise damage the chlorophyll. In autumn, when deciduous trees stop producing chlorophyll before dropping their leaves, the accessory pigments remain for a while. The yellow and orange you see in autumn leaves are the carotenoids that were always there, behind the green. The green fades, and the other colors are revealed. The red and purple of autumn leaves come from a different set of pigments, anthocyanins, which are actually newly synthesized in the autumn, perhaps as further protection for the leaf during its final days of function. Autumn color is not the death of the leaf. It is a last act of biochemical creativity. Now that we have our light-absorbing molecules in place, let's follow the energy. A photon arrives at a leaf. It enters the leaf tissue and reaches a chloroplast. Within the thylakoid membrane, it encounters a chlorophyll molecule. The chlorophyll absorbs the photon. The energy of the photon excites an electron in the chlorophyll. This electron, now carrying extra energy, is passed to the first molecule in a chain of electron carriers embedded in the thylakoid membrane. This chain is called the electron transport chain. And what happens along the chain is a beautiful piece of molecular engineering. The electron moves from one carrier molecule to the next. At each step, it releases a small amount of energy. That energy is used to pump hydrogen ions, protons, across the thylakoid membrane from the stroma into the interior of the thylakoid. The protons accumulate inside the thylakoid. The concentration gradient builds up. There are more protons inside than outside. And protons, like any molecule in a concentration gradient, want to move from high concentration to low concentration. They want to flow out, but they cannot cross the membrane freely. There is only one exit, a protein channel embedded in the membrane called ATP synthase. This is one of the most remarkable molecular machines in biology. As the protons flow through ATP synthase driven by the concentration gradient, like water through a turbine, the protein rotates, literally spins, about 150 times per second. And as it spins, it drives a chemical reaction that joins a phosphate group to a molecule called ADP to form ATP. ATP, adenosine triphosphate, is the universal energy currency of life. It is how cells store and transfer energy for almost everything they do. The spinning protein is manufacturing cellular fuel from a flow of protons, which was created by an electron cascade, which was triggered by a photon of sunlight. Sunlight, in a series of molecular steps, becoming the energy that powers a cell. But the electron that started this chain cannot be left hanging. After it has passed through the electron transport chain, giving up its energy to pump protons and generate ATP, it needs to be replaced in the chlorophyll. Where does the replacement electron come from? Water. This is perhaps the most important single reaction in the history of life on Earth. A cluster of proteins in the thylakoid membrane associated with what is called photosystem II does something remarkable. It splits water molecules. Two water molecules are broken apart. Their electrons are used to replace the electrons that chlorophyll has donated to the transport chain. The hydrogen ions from the water molecules contribute to the proton gradient, and the oxygen atoms are released as oxygen gas, O2. The oxygen you breathe, this is where it comes from. Not from some exotic chemical process. From water being split by light inside a chloroplast. Every breath you have ever taken was possible because of this reaction. The electron does not finish its journey after passing through the first electron transport chain. It is re-energized by a second photon capture event in a second set of chlorophyll molecules, photosystem I. After this second excitation, it passes through a shorter electron transport chain and finally arrives at a molecule called NADP+. Adding the electron and a proton to NADP+ creates NADF. NADF, along with the ATP made by the proton gradient, is the other key product of the like reactions. NADF is sometimes informally called hot hydrogen, a high-energy molecule that carries both electrons and hydrogen atoms in a form ready to do chemical work. So after the light reactions, we have two products: ATP, the energy currency, and NADDF, the high-energy hydrogen carrier. Both of them will now be used in the second stage of photosynthesis, a stage that does not require light at all. The second stage of photosynthesis is called the Calvin cycle, or the light-independent reactions, or sometimes the dark reactions, though this last name is misleading, because the Calvin cycle can happen in the light or the dark. It just does not require light directly. The Calvin cycle happens in the stroma, the fluid surrounding the thylakoids inside the chloroplast. Its purpose is to use the ATP and NADF made in the light reactions to build sugar from carbon dioxide, to take carbon dioxide from the air, an inorganic molecule, and use it to construct glucose, an organic molecule. This is carbon fixation, the taking of carbon from the atmosphere and incorporating it into living matter. It is one of the most fundamental processes of life. The Calvin cycle is named after the American chemist Melvin Calvin, who worked out its steps in the late 1940 and early 1950 using radioactive carbon-14 to trace the path of carbon through the reactions. He received the Nobel Prize in Chemistry in 1961 for this work. The cycle works like this. The key enzyme is RuBis-CO, ribulose 1, 5-bisophosphate, carboxylase, /oxygenase. It has the longest name of any common enzyme, and it is possibly the most abundant protein on Earth. Billions of tons of RuBis-CO exist in the world's plants, algae, and cyanobacteria. It is everywhere there is photosynthesis. RuBis-CO's job is to attach a molecule of carbon dioxide to a 5-carbon sugar called RuBP ribulose bisphosphate. The resulting 6-carbon compound immediately splits into 2 3-carbon molecules. These 3 carbon molecules are then processed using the ATP and NADF from the light reactions through a series of steps that ultimately produce glyceraldehyde 3-phosphate, or G3P. G3P is the key output of the Calvin cycle. It is the precursor to glucose. 2 G3P molecules can be combined to make glucose. Glucose can be used immediately as fuel, or polymerized into starch for storage, or used as a building block for other organic molecules, the cellulose of cell walls, the lipids of membranes, the biome, amino acids of proteins. Everything a plant is built from ultimately traces back to G3P produced in the Calvin cycle. Let's take stock of the whole process. 6 molecules of carbon dioxide from the air, 6 molecules of water from the soil, light energy captured by chlorophyll. The result, 1 molecule of glucose, plus 6 molecules of oxygen, released as a waste product. The glucose stores the energy. The oxygen escapes into the atmosphere. The carbon that was floating in the air as carbon dioxide is now part of a sugar molecule in a leaf. From there it may become part of the plant's structure, or be eaten by an animal, or be burned in a fire, or be incorporated into wood that lasts for centuries, or eventually be buried and become, over millions of tamba years, the carbon in coal or oil. The carbon cycle and the photosynthesis cycle are one and the same. Carbon moves from the air into life and back into the air in a loop that has been running for over 3 billion years. We should talk about how ancient this process is. Photosynthesis is not a recent invention of complex multicellular life. It is ancient, almost incomprehensibly ancient. The earliest photosynthetic organisms were bacteria, cyanobacteria, sometimes called blue-green algae, though they are not truly algae. They began producing oxygen through photosynthesis approximately 2.7 billion years ago. At that time, Earth's atmosphere contained very little oxygen. It was mostly nitrogen and carbon dioxide and methane. The emergence of oxygen-producing cyanobacteria was, in geological terms, a catastrophe. For the organisms that had evolved in the oxygen-free atmosphere, oxygen was toxic. The Great Oxidation Event, the dramatic rise in atmospheric oxygen caused by photosynthetic bacteria, wiped out vast numbers of those early oxygen-sensitive organisms. It was the first mass extinction. And it was caused by oxygen, the very thing we breathe and depend on. The organisms that survived were those that could tolerate oxygen, or even use it. Some evolved to use it in cellular respiration, the process that is essentially photosynthesis in reverse, burning organic molecules with oxygen to release energy. Cellular respiration and photosynthesis are complementary. Photosynthesis takes in carbon dioxide and releases oxygen. Cellular respiration takes in oxygen and releases carbon dioxide. Together, they cycle carbon and oxygen through the biosphere in an intricate, ancient, self-sustaining loop. The chloroplasts in plant cells were not always part of plant cells. They were once free-living cyanobacteria. About 1 to 1.5 billion years ago, a larger cell engulfed a cyanobacterium. But instead of digesting it, the cell kept it alive. The cyanobacterium continued to photosynthesize inside its host. The host cell benefited from the organic molecules the cyanobacterium produced. The cyanobacterium benefited from the shelter and raw materials the host provided. Over millions of years, the cyanobacterium lost the ability to survive independently. It became an organelle, the chloroplast. This event, called endosymbiosis, was one of the most consequential moments in the history of life. It gave rise to all the world's plants, algae, and other photosynthetic eukaryotes. Every tree, every flower, every vegetable, every blade of grass, all of them carry within their cells the ancient descendants of cyanobacteria that were swallowed and not digested over 1 billion years ago. The forest is, in a sense, a colony of ancient bacteria. The leaf is a collaboration. Not all photosynthesis is equally efficient. The most common form called C3 photosynthesis, because the first stable product as a 3-carbon molecule is used by the majority of plants, including wheat, rice, and most trees. But it has a weakness. Rubis-Co, the enzyme that fixes carbon dioxide, can also react with oxygen rather than carbon dioxide. This process, called photorespiration, is wasteful. It undoes some of the work of photosynthesis. In hot, dry conditions, when plants close their pores to reduce water loss and oxygen builds up inside the leaf, photorespiration can significantly reduce efficiency. Some plants have evolved a workaround. C4, photosynthesis used by maize, sugarcane, and many grasses, concentrates carbon dioxide in the cells around the key photosynthetic machinery, reducing the chance of oxygen interfering. This makes C4 plants significantly more efficient in hot, bright conditions. Sugarcane is among the most productive photosynthesizers on Earth, converting up to about 2% of the solar energy that falls on it into chemical energy. This sounds modest, but compared to the best human solar panels, which convert perhaps 20 to 25% of light into electricity, and considering that the plants are doing this continuously, growing, reproducing, and running all their biological processes on this captured energy, it represents an extraordinary biological achievement. Photosynthesis also underpins the fossil fuels that power our civilization. Coal, oil, and natural gas are the remains of ancient organisms, mostly ancient forests and ocean plankton, that photosynthesized millions of years ago. They captured carbon dioxide from the ancient atmosphere, built it into organic molecules, and when they died, were buried under sediment before decomposing. Over millions of years, heat and pressure transformed those organic remains into the hydrocarbons we now extract and burn. When we burn fossil fuels, we are releasing into the atmosphere the carbon that was taken out of it by photosynthesis hundreds of millions of years ago. We are running photosynthesis backwards, and much faster than it originally ran. The carbon that took millions of years to be buried is being returned to the atmosphere in decades. The implications of that reversal are the defining challenge of our time. As you lie here tonight, somewhere outside your window, if there is a tree, a garden, a park, a field, a forest anywhere near you, leaves are resting. In the dark, photosynthesis has paused. No new glucose is being made. The like reactions require light, but the Calvin cycle can run in the dark, using stored ATP and NADF, and so the work continues for a while after the light fades. By now, though, deep in the night, the plants are simply breathing, respiring, using the glucose they stored during the day, taking in a little oxygen, releasing a little carbon dioxide, resting, as you are. And when the light returns in the morning before you are likely to be awake, the chloroplasts will wake first. The photons will arrive. The electrons will leap. The protons will flow. The wheel of ATP synthase will spin. The water will be split. The oxygen will be released. And somewhere in the morning air, the breath you take when you wake up will be a little newer than the one you took last night. Thank you for spending this time with IQriously. Rest well. Breathe easy.